morning. My name is Lisette de Senerpont Domis, and I'm based at the Netherlands Institute of Ecology, where I'm head of a research group called AQUA. In addition, I'm Professor Smart Ecological Monitoring at the University of Twente. Today, I'm going to talk to you about remotely accessing mesocosms and solutions for more inclusive collaborations. In this webinar, I will explain you what mesocosms are, what they're used for, and how virtual and remote access to mesocosms can lead to more inclusive collaborations. So mesocosms. Mesocosms are somewhere in the middle between lab studies and ecosystem studies. They offer a near realistic complexity, but at the same time, due to their often large size, operating costs are quite high, and that also limits their replicability because we do not have often galore budget. I am funded or partially funded by the EU Infraia, so infrastructure project Aquacosm Plus. And Aquacosm Plus is really about networking mesocosm science. It's about the networked science of aquatic mesocosm specialists and about offering transnational access to their facilities. So it's really also about the mobility grant. It started as Mesaqua, and it then had only a couple of facilities there, and they were all marine. Then the follow-up program was called Aquacosm, where freshwater, for the first time, freshwater and marine mesocosm facilities were united. And then currently we're undergoing the third iteration, Aquacosm Plus, where we offer 13,000 access days and where we have both marine, freshwater, as well as running water facilities. And today I'm going to talk to you about highly controllable mesocosms. So not all these mesocosm facilities that are currently in Aquacosm Plus are highly controllable. Some are just merely buckets or outdoor cattle tanks, and others are very high-end and highly controllable. And examples of the very high-end, highly controllable mesocosms are the limnotrons, based at my facility, the Netherlands Institute of Ecology, and the planktontrons, which are, I would call a little bit smaller, but in some sense, more high-tech version of the limnotrons. And they're based at the University of Oldenburg. So the evolution of a mesocosm network. As part of this, there is a high level of control in these indoor mesocosms, but they often lack replications. For instance, at the NIOO, we only have 9 limnotrons. They often have some level of automated monitoring, although outdoor cosms such as IGB Berlin mesocosms have automated monitoring as well. They can offer virtual and sometimes even remote access. And I come back to the difference between virtual and remote access later on. But importantly, these indoor mesocosms offer less constraints on the introduction of pollutants and, for instance, invasive species. They also offer more opportunities of exposing them to more extreme events, such as climate change events. Importantly, the experiments that are happening indoor are not growth season dependent, and often because they offer a high-tech type of mesocosm facility, that often comes hand in hand with high-tech lab facilities nearby, such as in the NIOO we have high-tech molecular and chemical labs. So high level of control, why would you want to have that? So if we think about the limnotrons, which are the NIOO facilities, they were established already in 2001, and we were based then at a different location. So now we're based in Wageningen, and back then we were based near Amsterdam, Nieuwersluis. And back then they were built on these milk tanks, so very Dutch type of application. And they already, back in the day, offer a high level of control so that you were able to stratify the temperature. So we have upper and lower differential heating and cooling system. You were able to manipulate the water level, the water movement in different ways, an impeller versus an oscillating grid, or even a wind blower on the surface. We were able to manipulate carbon dioxide levels and the light regime. The limnotrons are about 1 meter high and about 132 centimeters in width, and they can contain almost 900 liters at max capacity. When you implement a sediment, which was possible more recently, then of course your water level will be less deep. What we see in the evolution of the limnotron since its establishment in 2001 is that we went from a system which was offered already quite a bit of control to a system which has a lot of controllable parameters. Now, not only can we manipulate the light regime, but we can also manipulate the light spectrum, and we can do this remotely as well. We already could manipulate the carbon dioxide levels, temperature, and stratification regime, but we can now also manipulate the flushing regime. So, to what extent what is the water residence time of the system? We can manipulate that, the water level, water movement, and now we can include sediment as well. Importantly, we are now able to manipulate also the way we sample. We have automated monitoring of light, temperature, oxygen, depth, and we have cameras mounted both in the water column as well as on top of the water column. And this automated monitoring can be remotely controlled. That means that if you are, for instance, in Australia, you can send a command to the local host computer in the sense that you want to have a depth profile made, or you can manipulate the temperature levels. What is important to keep in mind in these type of mesocosms is that they're often not, you wouldn't be able to run 100 mesocosms at the same time. So you have to start thinking of what is the experimental design of my system, and you have to perhaps move away from this more traditional type of ANOVA type of model towards a more regression design. And what we in the limnotrons often do is a replicated regression design, and where we look at, for instance, dose response relationships between a pollutant, for instance, along a gradient. Automated monitoring. What you see here on this dashboard is all the parameters that are constantly being logged from the limnotrons at a minute to second minute level. And why is it important to monitor these parameters at such high frequencies? For this, I'm going to take a little interlude and talk about the Nyquist theorem and the relationship between the Nyquist theorem and sampling frequencies. On the top panel, you can see an adequately sampled signal. So you see here sort of a sinoid wave, and you see that indicated by the dots that the sampling frequency is adequate to capture the signal. In the lower panel, you can see undersampling and what that does to your interpretation of the signal. So you see, for instance, that if we sample on these dots here, we see a signal which has a very much lower interval or frequency than our correct signal. And a good example, and perhaps more intuitive example, is the video sampling rate when you're videoing, for instance, the wings of a helicopter. If your sampling rate or your video sampling rate is too low, you actually don't see the wings moving. You see them standing still, and this is a good example of the Nyquist theorem. But to take this to the aquatic realm, we have to start thinking about generation times versus sampling frequencies. If we, for instance, look at zooplankton, they typically have a generation time of 1.5 to 5 days, whereas the sampling interval of most experimental facilities is on a weekly, bi-weekly basis. If we think about phytoplankton, they can have a generation times either lower, so 0.2 days, or much longer, depending on what species they are, and they have a similar sampling intervals as zooplankton, basically on a weekly, bi-weekly basis. If you then think about, for instance, a design where you have, for instance, a Baki design, a before and after intervention design, where you have sampling before an intervention, say a restoration measure, and after an intervention, then basically we're looking at the equivalent of evaluating the effect of fire on forest dynamics after 20 years. You wouldn't do that, right? So by sampling on a weekly, bi-weekly basis, we do not capture processes perhaps at the right rate. And what we're doing actually is missing transient events, and especially in the field of extreme climatic events, for instance, which are often transient events, then you can look at missing, for instance, phytoplankton blooms or cyanobacteria blooms. And this is a nice paper by Francesco Pomati and his team, where they showed that based on automated monitoring, they were able to capture a bloom, whereas on conventional monitoring, they were missing out on this bloom. Another argument for measuring high frequency is that measuring high frequency allows you to also calculate rates. And here we see an example of an experiment where we looked at dissolved oxygen rates, levels in the water column. And we calculated gross primary production and net ecosystem production based on these high frequency measures. And why is this important? And it is often causing the discrepancy between, say, more experimental biologists and modelers, because you need to have fluxes or rates to incorporate in your process-based models. Process-based models do not want a number of state variables, an additional number of state variables. They want to have more input on processes. So fluxes or rates are very important for our modeling exercises. Another thing which is very important about these indoor mesocosms, that they, as I said before, they offer less constraints on climate pollutant treatments. So we're looking at a future where a couple of big pressures are coming together, and I sometimes call this a perfect storm for water quality management. And importantly, there's an interaction between climate change on the one hand, which has these extreme climatic events, which are often transient. We have eutrophication, which is more of a press disturbance. And then we have emerging pollutants, which can act both as a press and a pulse disturbance. And these can cause disruption of aquatic food webs through a couple of processes, right? And I here display a couple of them, such as shifts in competitive advantage, phenological decoupling, stoichiometric constraints. So these are constraints due to the chemical imbalance of nutrients and disruption of chemical communication. To study these aquatic food web level effects, you need these type of large-scale facilities, right? On the one hand, you need high-level control. You need to be able to manipulate your pollutant levels, your climate regimes, and your eutrophication regimes. And on the other hand, you want also to have these near realistic type of food webs. Because just to give you a little bit more background, these are typically the type of organisms and processes we would study in these mesocosms. So importantly, we look at functional groups, but we also look at processes such as primary production, methane evolution and diffusive emission and decomposition. And we look at functional groups such as zooplankton, macrofauna, phytoplankton, bacteria, and periphyton, and sometimes even parasitic fungi. And that all accumulates in a number of high impact papers. However, I want to conclude this talk with talking more about why I think it's important to offer remote and virtual access to our facilities. And why? If you look at this map on the right side, you can see the amount of money as share of the GDP spent on research and development. And you see a large discrepancy between the global north and the global south, with the exception of Australia. So for large chunks of this world, it's virtually not possible to do the research they perhaps want to do due to their lower access to resources. Another argument for offering a remote or virtual access is the occurrence of these type of pandemics, as we see here now. We've been experiencing the COVID-19 pandemic, and did actually caused a large restriction on our mobility, right? We were not, and as a consequence, for instance, in the Aquacosm Plus project, we were not able to offer the transnational physical access that we were supposed to do under the grant agreement. And I think for another reason why you maybe want to have less mobility around the world can be climate change impacts, right? More and more people are considering climate change as a factor in restricting their flight movements, for instance. And by offering remote and virtual access, you would actually restrain or contain the number of flights you would have globally as a research team. So I want to explain a little bit of a difference between virtual versus remote access. And this is what the ESFRI roadmap tells you about this. They state that virtual access means free access to users provided through communication networks. The available services or resources can be simultaneously used by an unlimited number of users, and users are not selected. Virtual access typically concerns access to data and digital tools. In contrast, remote access is access to resources and services offered by the research infrastructure without users physically visiting the infrastructure facility. And it's in that sense very similar to physical access. The services or resources are not unlimited, so they are limited, and a competitive selection is required. I give examples of the things we do both on the virtual access front as well as on the remote access front. A remote access in our sense is also remote control. And as I explained before in a less detailed figure, that means that a remote user through an internet data link can log into our local computer where we host SpecView as our, well, SpecView is basically our local data harvesting program. And through a Modbus over Ethernet communication with a control unit for each limb neutron, we can then, if we were in Australia, we can sample the water column for these autonomous measurements, such as pH, oxygen, depth, and light. So that is what we have at the moment for remote control. On the other hand, we also have virtual access, and that is that is unlimited, right? So that for that you do not have it's open. You do not have to have a login procedure in place. And what it basically does is we have a mesocosm. The mesocosm sends information to the local server. The local server then sends this information to the cloud database, and the cloud database subsequently sends this to a cloud-based data portal. And this allows you to look at both present data. So if you have an actual mesocosm experiment running, you can just on a near real-time basis, you can look at high frequency data of a number of parameters: depth, temperature, light, and oxygen. But in our case, you can also look into the past by selecting, for instance, different time windows and then download data. So this is what we offer in terms of virtual access. Then one of the latest features of the limnotrons is through actually looking at our limnotrons remotely. So what we have right now is both cameras installed within the water column as well as on top of the water column. And here you see a display of the camera that is mounted within the water column. And what it does, it allows us to visually track primary producers. This experiment was about blanket weed, and these are these filamentous algal beds. And they start at the bottom, and then gradually they move upwards in the water column. You can see similar processes, for instance, with benthic cyanobacteria, such as Formidium, for instance. They also start at the bottom and then move towards higher up in the water column. And what this visual tracking allows us to do is, for instance, through image analyzing the field of view, we can get a sort of biomass estimate, but it also allows us to track where these primary producers are in the water column. So here we see, for instance, a picture of the camera mounted on top of the limnotrons, where you see that these blanket weed beds have ascended near the surface. So this is the latest feature of our mesocosms. Well, I hope to have given you some sort of flavor in what we do in this line of research in terms of virtual and access and remote control. And I'm sorry to not be here in person. We had government representatives visiting our lovely institute today. But if you have questions, don't hesitate to contact me either through email or using my Twitter handle. And I want to thank you for your attention, and hope to see you somewhere in the future.